Dear student, I am Dr. 𝗓𝐴𝐻𝐵𝐞, Professor of Biochemistry. So in this lecture, I will be going over de novo synthesis of purine nucleotides. Let's get into the details. Now the purine synthesis can be broadly classified into three major ways that we can synthesize purines in our cells in a decreasing order of importance. One is synthesis of purines from amphibolic intermediates, that is called as the de novo biosynthesis of purine nucleotides. Second is phosphoribosylation of free purine bases that are coming from cell turnover or from the food. That is basically referred as 𝗟𝐵𝐴𝐺 pathway. And the third is phosphorylation of purine nucleosides coming from cell turnover to make nucleotides. That is another 𝗟𝐵𝐴𝐺 pathway. So basically we have two 𝗟𝐵𝐴𝐺 pathways and one de novo biosynthesis of purine nucleotides. So in this particular lecture, we are going to go into de novo biosynthesis of purine nucleotides. Now, a purine ring, first we'll have to see what are the atoms that are present in the purine ring and what are the donors of these atoms that are used to make this purine ring in a de novo biosynthesis of purine nucleotides. So let's look into a free purine base and see what are the different atoms which are present here. So free purine base has got 9 atoms and they are numbered in anti-clockwise manner as it is, like it started from N1, nitrogen 1, and then we have C2, N3, C4, C5 and C6, and then we have N7, C8 and N9. Now let's look at the donors of each one of these atoms present in a purine ring. So N1 is donated by amino acid aspartate, and carbon 2 is coming from N10 formyl tetrahydrofolate. Then nitrogen 3 and nitrogen 9, it will come from amide nitrogen of glutamine, that's a side chain nitrogen of glutamine. And then C4, C5 and N7, this entire thing will come from glycine, that is 2 carbons and 1 nitrogen present in glycine, almost entire glycine is incorporated here. And then C8, carbon 8 will come from N5, N10 methynyl tetrahydrofolate. So these are the donors of all the 9 atoms that are present in purine ring. Now let's look into overview of de novo synthesis of purine nucleotide. So this particular figure is showing you the overview of de novo biosynthesis of purine nucleotide. Note that most of the purines in our body are synthesized predominantly in the liver and then they are transported out of the liver to other extrahepatic tissues. Alright, so there are 13 reactions in de novo biosynthesis of purine nucleotide. So with those 13 reactions, it will be giving only up to inosine monophosphate. So up to the formation of purine mononucleotide, that is inosine monophosphate, it needs 13 reactions and 7 ATP. So it's energetically, it needs a lot of energy to make inosine monophosphate and also it takes a lot of time. So de novo synthesis of purine nucleotides, it takes time and energy for a cell to make a purine nucleotide from the scratch. Remember that. And that AMP, it will go into different direction. It can go towards adenosine monophosphate and then adenosine diphosphate and then adenosine triphosphate to go towards RNA molecule. Or adenosine diphosphate can undergo ribonucleotide reductase reaction where a ribose sugar is converted to deoxyribose sugar to make deoxyadenosine diphosphate, which will be further converted into deoxyadenosine triphosphate to take that into DNA formation. This is how the adenosine monophosphate goes. Or IMP going into GMP, guanosine monophosphate side. So guanosine monophosphate to guanosine diphosphate and to guanosine triphosphate going into RNA side, or guanosine diphosphate undergoing ribonucleotide reductase reaction making deoxyguanosine diphosphate and thereby deoxyguanosine triphosphate, and that is taken into DNA formation. So like this, 13 reactions, 7 ATPs making inosine monophosphate. Later it will diverge into 2 directions towards the AMP and GMP, and from there the necessary nucleotides for DNA, purine nucleotides for DNA and RNA can be synthesized. So, some of the, like all the donors that are participating in de novo biosynthesis of purine nucleotides are shown in this particular figure. So, sequentially they are getting into the reaction and they are going to donate each of their atoms to build a purine nucleotide. Remember that. And also note that ribose-5-phosphate, which is coming from pentose phosphate pathway, is the one that initiates the purine nucleotide biosynthesis, because ribose-5-phosphate, it is going to condense with the ATP to make PRPP. PRPP is phosphoribosyl pyrophosphate, and this phosphoribosyl pyrophosphate is the one that initiates purine nucleotide biosynthesis and also as well as pyrimidine nucleotide biosynthesis. Remember that. So, formation of PRPP is essential here for both purine and pyrimidine biosynthesis. So, you need to remember that point. Now, let us look at how exactly PRPP, that is 5-phosphoribosyl pyrophosphate. So, that is phosphoribosyl pyrophosphate, how exactly it is made. So, PRPP is the necessity in purine nucleotide biosynthesis, because the purine ring, entire purine ring, it is built over PRPP. Phosphoribosyl pyrophosphate is the initial requirement over which purine ring is built from scratch. Okay, so how PRPP is synthesized? You must remember how this happens. So, because it is the one that is most essential to initiate purine nucleotide biosynthesis. So, PRPP is synthesized from ribose-5-phosphate, which is coming from pentose phosphate pathway. So, ribose-5-phosphate taking a phosphate, 2-phosphate from ATP. ATP is broken down into AMP here, and those 2 phosphates is attached to the ribose-5-phosphate to make 5-phosphoribosyl pyrophosphate. That's what is PRPP. And this entire reaction is catalyzed by enzyme PRPP synthetase enzyme. PRPP synthetase is the enzyme that helps in the formation of PRPP. So, once we get PRPP, so this PRPP will be used to build a purine nucleotide over it. Okay. Now, other than PRPP being a precursor for purine nucleotides, so it is also needed for synthesis of pyrimidine nucleotide biosynthesis. Along with the pyrimidine nucleotide biosynthesis, PRPP is necessary for NAD, nicotinamide adenine dinucleotide formation, nicotinamide adenine dinucleotide phosphate formation, histidine synthesis, and also in the synthesis of tryptophan. So, in all these processes, PRPP is necessary. So, that's the other uses of PRPP other than it is participating in purine nucleotide biosynthesis. And also note that PRPP, as I was saying earlier, it is the positive modulator of both purine nucleotide biosynthesis and also pyrimidine nucleotide biosynthesis. So, to synthesize both purines and pyrimidines, there is a need of PRPP. Remember that. Okay. And intracellular concentration of PRPP is subjected to meticulous regulation because it is necessary for synthesis of both purines and pyrimidine nucleotides. Now, let's look at what happens once we get PRPP, that is phosphoribosyl pyrophosphate. Now, the phosphoribosyl pyrophosphate undergoes a second reaction in de novo purine biosynthesis. So, the first reaction we have seen in the previous slide. So, the first reaction is it is conversion of ribose-5-phosphate into PRPP done by PRPP synthetase. Okay, that's the very first reaction in de novo biosynthesis of purine ring. Now, the second reaction is catalyzed by PRPP glutamyl amidotransferase or PRPP amidotransferase enzyme, or it is written as PRPP glutamyl amidotransferase, which is called as glutamine phosphoribosyl amidotransferase. So, there are two names here in the second enzyme in purine biosynthesis, purine nucleotide biosynthesis. So, it is PRPP glutamyl amidotransferase, simply sometimes written as PRPP amidotransferase, or it can be written as glutamine phosphoribosyl amidotransferase. So, what it does? It's going to take PRPP, PRPP is acting as a base or a support over which a purine ring, each atom is brought to make a purine ring here. So, what is done in this second reaction? the glutamine, the nitrogen, amide nitrogen of glutamine is taken and it will be added on to PRPP to make that N9, that is the 9th nitrogen present in a purine base. It is the first one that is added, which is coming in the form of glutamine. Amide nitrogen of glutamine provides that nitrogen, that is the 9th atom in a purine base. Remember that. That's the second reaction, and this is the most regulated reaction in purine nucleotide biosynthesis. That also it is a rate determining step in purine nucleotide biosynthesis. Once we get 5-phosphoribosylamine, this 5-phosphoribosylamine, it will undergo 3 to 11 reactions, and so at the end of 11th reaction we are going to get inosine monophosphate. Okay, so ribose-5-phosphate to PRPP, that's number 1 reaction, that is done by PRPP synthetase enzyme. Then the PRPP to 5-phosphoribosylamine, where 9th atom of purine ring is added, coming in as amide nitrogen of glutamine, done by PRPP glutamyl amidotransferase, which is also called as glutamine phosphoribosyl amidotransferase. So it's a second reaction. From there, 3 to 11 reaction, where each and individual donors, they will come in and donate their atoms, just like what is shown in this particular figure. As you see, let me just briefly take you into that. So this is number 1 reaction. Number 2 reaction. In the number 3 reaction, glycine comes in, which will give C4, C5 and N7. Number 4 reaction, where N10 formyl tetrahydrofolate comes in and it gives carbon 8 of purine base. And then number 5th reaction, where one more glutamine comes in and gives N3 of purine ring, and then there will be a ring closer there. And number 6 reaction, carbon dioxide coming in and give carbon number 6 in a purine ring. Number 7 reaction, aspartate coming in gives that nitrogen 1, very first nitrogen coming from aspartate. And number 8 reaction here, N10 formyl tetrahydrofolate coming in and giving that carbon 2 of a purine ring. And then inosine monophosphate. Inosine monophosphate made, and that inosine monophosphate can be going into AMP by 2-step reaction, or inosine monophosphate going to GMP by 2-step reaction. So sequentially like this, so each and every atom is brought in and it will be added by 3 to 11 reaction, ultimately making inosine monophosphate. So once we get inosine monophosphate, as we have seen in the previous figure, so inosine monophosphate, this is inosine monophosphate, it will go towards adenylosuccinate, and adenylosuccinate going towards adenosine monophosphate. So there are 2 enzymes involved here. One is adenylosuccinate synthetase enzyme, and other is adenylosuccinate lyase enzyme. So with the help of adenylosuccinate synthetase and adenylosuccinate lyase enzyme, so inosine monophosphate is ultimately taken towards adenosine monophosphate formation, that is AMP. Okay, on the other side, IMP dehydrogenase enzyme taking inosine monophosphate into xanthosine monophosphate, and then by GMP synthetase enzyme it will be converted into guanosine monophosphate. So like this, IMP, it is diverging into 2 sides. One is it is going towards AMP formation, other is it is going towards GMP formation. So while going towards AMP formation, it is going through adenylosuccinate synthetase enzyme catalyzed reaction, and while going to GMP formation, it is going to go through IMP dehydrogenase reaction. Okay, so there are drugs like 6-mercaptopurine. It inhibits PRPP glutamyl amidotransferase. It is inhibiting adenylosuccinate synthetase, and also it inhibits IMP dehydrogenase enzyme. So overall, 6-mercaptopurine, it is inhibiting a purine nucleotide biosynthesis at 3 enzyme levels. So that's the importance of 6-mercaptopurine here. So it is going to first act on rate determining step in purine nucleotide biosynthesis, that is PRPP glutamyl amidotransferase, and then it is going to block this enzyme adenylosuccinate synthetase, and also it's going to block this enzyme IMP dehydrogenase. Overall, it is going to decrease formation of AMP and GMP. Both the purine nucleotides can be decreased. Okay, so if all goes well and no inhibition, so at that time so you get AMP and GMP. So AMP can further convert it into ADP, ADP into ATP. On the other side, GMP can be converted to GDP, and that GDP can be converted to GTP. So this is how ATP and GTPs can be made, and they can be taken towards RNA formation. So if there is a need for DNA synthesis, so AMP converted to ADP, and ADP will undergo ribonucleotide reductase reaction. We are going to see that in the next lecture, to make a deoxyadenosine diphosphate, which is further converted to deoxyadenosine triphosphate to go into DNA. Similarly, GMP to GDP, once you get GDP undergoes ribonucleotide reductase reaction to make DGDP, and this DGDP converted to DGTP, and that can also go towards DNA formation. So this is how purine nucleotides are supplied for both RNA and DNA synthesis. So here is the summary of purine nucleotide biosynthesis. So the first reaction is PRPP synthetase reaction, where PRPP is made. Second reaction is a rate determining step and most regulated step in purine nucleotide biosynthesis, that is formation of 5-phosphoribosylamine done by PRPP glutamyl amidotransferase. After that, 3 to 11 reaction. So we are going to get IMP, each atoms coming in and donating their atoms for purine nucleotide biosynthesis. IMP to AMP and IMP to GMP by adenylosuccinate synthetase and IMP dehydrogenase, and later they will get ATP or GTP for RNA, DGTP and DATP for DNA synthesis. Now, the drugs that are inhibiting purine nucleotide biosynthesis, 6-mercaptopurine, as I already told you, it is going to inhibit 3 enzymes in purine nucleotide biosynthesis. That is the rate determining enzyme, which is PRPP glutamyl amidotransferase, and then adenylosuccinate synthetase and IMP dehydrogenase. These are the 3 enzymes inhibited by 6-mercaptopurine. Then the sulfonamides, they are going to inhibit formation of folic acid by inhibiting dihydroteride synthetase enzyme, thereby inhibit folic acid synthesis in the bacteria. Methotrexate, aminopterin and related compounds, they are going to target tetrahydrofolate formation, THF, because they are going to inhibit DHFR, dihydrofolate reductase enzyme, thereby they block conversion of dihydrofolate into tetrahydrofolate, thereby tetrahydrofolate derivatives which are used in purine and pyrimidine biosynthesis cannot be coming here, because tetrahydrofolate derivatives which are going to give C2 and C8 atoms of purine ring. So that's how they are going to inhibit purine nucleotide biosynthesis. And also methotrexate, they are going to decrease the conversion of DUMP into DTMP in pyrimidine biosynthesis also. Glutamine antagonists, so they are going to decrease, like azacitidine, they are going to decrease the availability of glutamine for purine and pyrimidine nucleotide biosynthesis. Remember that. Coming to regulation of purine nucleotide biosynthesis, there are 4 regulatory enzymes in purine nucleotide biosynthesis, and one is the most important enzyme, and it is the most regulated enzyme in purine nucleotide biosynthesis, and also it is the regulated enzyme. Okay, so rate determining enzyme also, that is glutamine phosphoribosyl amidotransferase, which is also called as PRPP glutamyl amidotransferase. Okay, it is going to catalyze second reaction in purine nucleotide biosynthesis. So all 3 nucleotide forms, that is GMP, GDP, GTP, AMP, ADP, ATP, all 6 of them will have a feedback negative effect on glutamine phosphoribosyl amidotransferase. Now the other enzyme that is modulated by the feedback inhibition process is GDP, and ADPs will have a negative effect on PRPP synthetase enzyme. Remember that. And immediate negative effect can be seen in purine nucleotide biosynthesis, that is GMP will have a negative effect on IMP dehydrogenase, whereas AMP will have a negative effect on adenylosuccinate synthetase enzyme. Okay, so these are feedback negative effects here. So whereas PRPP, elevated levels of PRPP will have a positive effect on PRPP glutamyl amidotransferase. So that type of regulation is also possible here. Now there is a cross regulation in the conversion of IMP to AMP and IMP to GMP, because whenever there is a high levels of GTP, that GTP will promote conversion of IMP to AMP. So higher levels of GTP will take IMP towards AMP formation and ultimately making ATP. So whenever there is higher ATPs and lower GTPs, at that time higher ATPs will have a positive effect on conversion of xanthosine monophosphate into guanosine monophosphate, basically taking IMP into GMP. Okay, so this kind of cross regulation will make sure that both ATPs and GTPs are synthesized in equal concentration so that DNA and RNA synthesis can be done. So this is all about de novo purine biosynthesis and regulation of purine biosynthesis. Thank you.